Autumn yields a massive harvest of vegetables, but they come with a biological ticking clock. Left alone, fresh produce rapidly breaks down, consumed by airborne microbes until it collapses into rot long before the winter freeze. To survive the lean months, humans had to find a way to stop this decay. Over centuries, two opposite biological strategies emerged to solve the exact same problem: eradication and cultivation. The eradication approach is centered on total microbial suppression. Methods like industrial canning use high heat, while standard pickling relies on soaking the food in harsh external acids to stop the natural breakdown process. Submerging a cucumber in a jar of vinegar immediately changes the pH of the environment. This artificial flood of acid prevents any airborne bacteria or yeast from taking hold. This effectively prevents rot, keeping food safe for months. But the resulting jar is sterile. Because it kills all microorganisms, the food is biologically dead, offering no living immune benefit. However, cultivation takes a very different path. Traditional homemade kimchi is packed into dark ceramic crocks and left outside, far from the sterilized conditions of a factory line. Instead of introducing an external acid like vinegar to kill microorganisms, this process relies entirely on a precise mix of salt, water, and thyme. Raw Napa cabbage is handled closely, massaged with spices, and buried in its own natural juices. Rather than preventing microbial life, this process intentionally invites wild bacteria from the soil, the air, and the hands of the makers to thrive. Leaving raw vegetables covered in wild bacteria at room temperature is generally the baseline for food spoilage. Yet consuming homemade kimchi delivers a potent dose of living, immune-boosting microbes. The fermentation jar functions as a rigorous evolutionary filter, transforming raw vegetables into a stable, active ecosystem. Once the cabbage is packed tight and sealed, the environment becomes oxygen-deprived. The heavy dose of salt draws out moisture, creating a highly concentrated brine. The naturally occurring microorganisms trapped inside have only one available fuel source: the complex carbohydrates and sugars within the cabbage leaves. As they break down these sugars through lactic acid fermentation, the microbes excrete organic acids directly into the brine. As the population of feeding microbes grows, the concentration of lactic acid rises, driving the internal pH level down into a highly acidic range. Unlike the instant flood of vinegar in pickling, this is a slow internal acidification generated by the bacteria themselves. The combination of zero oxygen, high salinity, and dropping pH becomes a lethal pressure cooker for common spoilage bacteria and dangerous pathogens, which cannot survive the acidic stress. By the third week, the dropping pH has eliminated the non-acid tolerant competitors, leaving a concentrated population of high-performance bacteria. The primary survivor of this fermented gauntlet is a lactic acid bacterium called Lactoplantibacillus plantarum. To endure the jar, L. plantarum utilizes a robust biological toolkit, synthesizing organic acids and metabolites like GABA, an inhibitory neurotransmitter. But its most effective survival mechanism is the production of bacteriocins. Bacteriocins are antimicrobial proteins that L. plantarum releases to hunt and destroy competing pathogenic bacteria, effectively dissolving their cell walls to secure its territory. This genomic map shows the DNA structure of L. plantarum isolated from kimchi. The colored arrows represent secondary metabolite gene clusters coding for bacteriocin proteins. These gene sequences prove L. plantarum is a specialized defender, genetically equipped to produce chemical weapons to dominate its environment. When a person eats fermented kimchi, the perspective shifts from the jar to the human body. A single portion deploys millions of these battle-tested microbes directly into the digestive tract. They immediately face a new set of lethal barriers: the gastric acid of the stomach and the corrosive bile salts of the upper intestine, which destroy most ordinary dietary bacteria. But because L. plantarum evolved to survive the extreme acid and salt of the kimchi jar, it possesses the specific biological armor required to pass through human digestion intact. Upon reaching the lower intestines, the surviving bacteria begin the process of colonization. They demonstrate a high capacity for cellular adherence, clustering together and physically anchoring themselves to the ridges of the intestinal wall. Once anchored, this colony uses its bacteriocin weapons to proactively shield the intestinal surface from incoming pathogens,
balancing the gut microbiome. By physically occupying the intestinal wall, these colonies create a biological barrier that limits the passage of toxins into the bloodstream. This reduction in gut leakage stabilizes the internal environment, allowing the immune system to shift its resources toward the production of natural antibodies. While standard pickling preserves food by creating a sterile void, homemade kimchi demonstrates that long-term health is supported by cultivating living, battle-tested ecosystems that defend the body from the inside out.